Welcome back. We're going to next pick up with where we left off in Learning Objective 1, and we're going to pick up with the loop of Henle. Now we have a thin descending limb of the loop of Henle that is impenetrable to solutes, but is penetrable to water. And we're not going to go over the mechanisms by which water moves through that, other than it involves aquaporin channels. In the thick ascending limb, which is where our focus is going to be next, the thick ascending limb is the opposite. It is impenetrable to water and is penetrable to a lot of different solutes. And the solutes I want to focus on in our lecture are written here, and they are sodium, potassium, calcium, and magnesium. Now, if you remember back from that first early on slide that showed the entire nephron and all of the reabsorptive processes, we said that the majority of solutes were reabsorbed in the proximal tubule, with the exception of magnesium. And I mentioned we would talk about magnesium again later. Well, here's where magnesium shows up again. Most magnesium, unlike the others like sodium and potassium and calcium, most magnesium reabsorption takes place in the thick ascending limb, and that's why it's depicted here, 60%. Everything else is less than the proximal tubule. In order to talk about the function of the thick ascending limb and how these 4 different solutes are reabsorbed, we are going to focus on the NKCC2 channel, which is sometimes written as Na2Cl K channel. It can also be written as the 1 sodium, 2 chloride, 1 potassium channel, meaning that whenever something is moving through this co-transporter, the NKCC2, it involves all 3 of these solutes moving together, such that 1 sodium will use the channel at the same time as 2 chlorides, at the same time as 1 potassium. And you'll note that 1 sodium is 1 positive charge. Chlorides, if you have 2 chlorides, that's 2 negative charges, and 1 potassium is 1 positive charge. So there are 2 positive charges and there are 2 negative charges that are entering using this transporter here, meaning whenever these solutes use this co-transporter, it is an electrically neutral event because 2 positives and 2 negatives cancel each other out, right? So when we have 1 sodium, that's 1 positive. When we have 2 chlorides, those are 2 negatives. And then 1 potassium again, that's 1 positive. So it's an electrically neutral event. That's going to play an important role pretty soon. Let's begin with orienting ourselves with the graphic here depicting cells in the thick ascending limb. On the right is the urine side. This is the tubular lumen, which is why it is depicted as a yellow tube. On the left is a red tube depicting the vasculature. Reabsorption would take place by solutes moving from the tubular lumen side into the vasculature. As I mentioned on the previous slide, the function of the NKCC2 co-transporter is vitally important for the reabsorption of solutes in the thick ascending limb. It is depicted here on this graphic at this location. Kind of a wonky looking line there. So solutes that are moving in, they are co-transported in at the same time. These solutes are sodium, chloride, and potassium. They enter into the cell using the NKCC2. And then once inside the cell, they can navigate through the cell and they have to figure out their destinies, if you will. Speaking specifically first about sodium, if we track sodium, once it's inside the cell, it will exit the cell across the basolateral membrane. Remember, this is the basolateral side on this side, and the apical side is over here on the right. It crosses the basolateral side using the sodium-potassium ATPase, which is right here. It exits the cell and then gets into the blood vasculature. What about the fate of chloride? So chloride enters in via the NKCC2, navigates across the cell, and then it exits the cell using a specific chloride transporter or a chloride channel, if you will. And that's how it exits the cell across the basolateral membrane and then gets back into the blood. Potassium is the last solute to discuss. Potassium has a very specific kind of dual destiny. Potassium gets inside the cell and it can do one of two things. Some potassium will continue to navigate across the cell and then exit the cell using potassium channels or potassium transporters. If this occurs, then the potassium that does move out across the basolateral membrane gets moved into the blood vasculature and it gets reabsorbed. But there is another fate. There are two roads that potassium can take. And the second road is that it can move back out the way in whence it came and move out again back across the apical membrane using something called an ROMK channel. ROMK channels are the channels by which potassium will what we call back leak. Potassium, every time potassium enters using the NKCC2, some potassium have the destiny of getting reabsorbed, and some potassium have the destiny of just falling right back out from whence it came. What happens then is that for every time these solutes get reabsorbed, some potassium builds back up inside the tubular lumen. And every time the channel turns over, some potassium just builds up, and it keeps doing that. And the more it does that, the more and more it does that, more and more potassium back leaks. Now note that the more potassium that back leaks, the more positive charges build up. So as our stuff ascends up the thick ascending limb, as we climb higher and higher, the tubular lumen grows more and more positive because of all this kind of continued back leak of potassium. So since now we're getting a net positive charge building up in the tubular lumen, please note that there are other things that exist in the tubular lumen that are also negative, namely magnesium and calcium, which are also positively charged. As we ascend up the thick ascending limb, the back leak of potassium, back leaking through these ROMK, just in case you are curious, stands for renal outer medullary K channel. So back leaks out this ROMK channel, builds up positive charge. The positive charge that builds up acts as a natural repulsor, I don't think that's a word, acts as a natural repellent, there we go, to magnesium and calcium, which are also heavily positive charged. As a result, magnesium and calcium will exit the area using a paracellular route. Remember that paracellular route is the route between cells. And that is why we call this a passive diffusion, meaning it is passively getting diffused or passively moving away from all this positive charge to get away from something that is repelling it. And that, in a nutshell, is how our important solutes, including sodium, chloride, potassium, magnesium, and calcium, get reabsorbed in the thick ascending limb. Next, we're going to go into the distal tube. Now we have moved on to the distal tubule. Let's orient ourselves to some terminology. When I talk about the distal tubule as a physiologist, the distal tubule is basically everything at the distal convoluted tubule and later. So it's the most distal part. But specifically, we can talk about the distal convoluted tubule, the cortical collecting duct, and the medullary collecting duct, and then the on towards the bladder using the ureter. When it comes to the distal tubule, these are the important kind of high yield notes to keep in mind. The least amount of solutes are reabsorbed here. And by amount, I mean essentially the molecules or the number of actual solutes. So the number of, say, sodium or the number of potassium ions that are reabsorbed take place here. Where are they most reabsorbed? Well, they're most reabsorbed in the proximal tubule. Try to remember that. It's kind of confusing because the distal tubule is what we talk about the most because the most important site of regulation of reabsorption or secretion processes. And as a result, there is a variable secretion and or reabsorption depending on the body conditions at the time. So our percentages are over here on the left: sodium, calcium, magnesium. The least amount, that's why there's only 5% or so, are reabsorbed here. Sodium as well. And then when it comes to water and urea, those can either be reabsorbed in variable rates or variable amounts. There are 3 cell types to keep straight in the distal tubule: the principal cells, alpha intercalated cells, and beta intercalated cells. And then finally, there are 2 hormones to really keep straight as far as I'm going to talk about, and they are aldosterone and ADH. But you could also talk about PTH as well, and probably some others that we won't go into. Next, we're going to go into talking about how these solutes are reabsorbed. Here are the 3 cell types I just got done mentioning that are important in the distal tubule: the principal cells, alpha intercalated cells, and beta intercalated cells. Now, each individual cell, like a principal cell, can either reabsorb or it can secrete. And the things that it reabsorbs or secrete are very specific, and each cell type is very specific. This table is something to definitely memorize. I don't usually really recommend a lot of really hard, serious memorization, but this is definitely one table to memorize. It will really help you out as you go through this. As we continue on, we're going to look at reabsorption in principal cells. We'll look at secretion in principal cells, and then reabsorption in alpha and beta intercalated cells, and then secretion. So these are all separate processes. I will focus the most on sodium, water, and potassium, and talk a little bit about bicarbonate. I'll talk about chloride, but only a little bit about hydrogen. So you're going to learn all of these hydrogen bicarbonate processes in your acid-base physiology as a separate lecture, because it really is kind of not only its own system, and it is integrated with everything else, but believe me, it can definitely warrant its own time and attention. Therefore, kind of understanding this now, keeping these 3 cell types straight, what they reabsorb and what they secrete, will really help you out as you get into that acid-base physiology. We will begin with the earliest most portion of the distal tubule, and that is the distal convoluted tubule. Here, this section of the distal convoluted tubule and the epithelial cells that are located here are relatively impermeable to water. However, it is permeable to solutes. Therefore, that means that as solutes get reabsorbed and water stays behind, that there is a dilution that will occur of the tubular fluid. And that will become important later. Here's our graphic again. The apical side is on the right, the basolateral side is on the left. To reabsorb, we're going to focus specifically on sodium and chloride. To reabsorb sodium, to cross across the apical membrane, we're going to use a very specific code transporter called the NCC. The NCC. NCC stands for the sodium chloride channel. It's kind of or sodium chloride cotransporter. It's pretty straightforward. But this is the only place in the nephron that the NCC exists. The NCC is also known by the name thiazide sensitive sodium and chloride cotransporter over channel. although technically it is a cotransporter. This means that this is the channel that is sensitive to a diuretic class called thiazides, which you are going to learn all about in next year's CPR2 class when you learn about diuretics. For now, let's focus on the fate of sodium chloride. The sodium will get reabsorbed from the urine side, enter into the cell using the NCC, navigate across the cell, and then like the other situations, get moved out of the basolateral membrane using the sodium-potassium ATPase, and from there it'll move into the blood. Chloride will move using just chloride channels. In the same area of the distal convoluted tubule that occur where the NCC occurs, there is another important receptor and channel that is responsible for the reabsorption of another important solute. So I mentioned before we were going to talk about sodium and chloride. Now we're going to add calcium into the mix. Calcium is reabsorbed in this area of the nephron using something called a TRPV5 channel. Let's do a little throwback to neuroendocrine. If you may or may not remember, there was another TRP channel that we talked about called TRPV1, TRPA1, and then TRPM8. Those are all involved in pain sensing. Well, here's another TRP channel. Rather than TRPV1, it's now TRPV5. Now, that doesn't mean that it is involved in pain. It just means it functions in the same way. It is a ligand-gated ion channel. We're not going to talk about the ligand in this context. However, it just means that when the ligand binds the channel, the pores open and an ion is allowed to move through the pore. In this case, the ion is calcium. So calcium is reabsorbed here. Similar to our other situations, let's talk about the fate of calcium. How does calcium move across the intracellular compartment, reach the basolateral membrane, and then get out again? Well, it gets out again using something called an NCE, or a sodium-calcium exchanger, meaning that for calcium to get out, a sodium has to get pumped back in. We will begin with talking about principal cells. Principal cells are responsible for sodium and water reabsorption and potassium secretion. Let's talk specifically about sodium first. The lumen side of this graphic would be over here on the left. So this is the apical membrane and this is the basolateral membrane on the right. So the urine side is on the left and then the blood side is on the right. Reabsorption then therefore means we're going to move from left to right across this graphic. Beginning first with sodium. Sodium has to cross the apical membrane. Now earlier in the nephron, like the proximal tubule and the thick acini limb, sodium entered either with a buddy or as a cotransporter. Regardless, all those different cotransporters transported sodium with something else. This is a different type of situation now. Rather than a cotransporter, we have what we call an ENaC. ENaC stands for epithelial sodium channel. So this epithelial sodium channel is essentially just a channel that is dedicated specifically to sodium located in an epithelial cell, so ENaC. The sodium will diffuse down its concentration gradient from the lumen side into the cell using this ENaC. And then the fate of sodium is to cross the basolateral membrane using the sodium-potassium ATPase, which is what this over here on the right is depicting. This is sodium-potassium ATPase. Meanwhile, potassium is secreted and essentially is going to move in the opposite direction, meaning potassium moves from the blood across the cell this way towards the urine side. So that's why we call it secretion. It's moving in the opposite direction as reabsorption. Reabsorption is up here. Potassium enters the cell using the sodium-potassium ATPase, navigates across the cell, and then exits the cell using a potassium channel. So for every 1 sodium that is reabsorbed here, a potassium is secreted. This cell and the action of these transporters and the channels are responsible for the fine adjustments in tubular fluid sodium concentration. And when we talk about aldosterone and its function in terms of stimulating sodium reabsorption acts on principal cells. It does so by stimulating this epithelial sodium channel. So the action of aldosterone is on these epithelial sodium channels. This is another view of the principal cells using the graphical layout I have been using the rest of the lecture. And again, these are our principal cells. These components should look familiar. We just got done talking about them in the last slide. So here is the ENaC channel. So when sodium is in the tubule, it enters using the ENaC, navigates across the cell, and then exits using the sodium-potassium ATPase. Now remember, the sodium-potassium ATPase, meanwhile, is bringing in a potassium every time a sodium leaves. This potassium has a fate, and that is to leave the cell again using those ROMK channels. We talked about that earlier, those renal outer medullary potassium channels. And the action of aldosterone, which is what the purpose of this little lightning bolt is it's going to zap the ENaC channel. So it increases the expression of those ENaC channels, meaning it literally is going to increase the transcription and translation of more ENaC channels and just plug them in to this side of the membrane, meaning that sodium can just enter in like just over and over again, reabsorb lots of sodium. Meanwhile, aldosterone also leads to an increase in expression of the sodium-potassium ATPase because this ATPase needs to continue to function and go into overdrive in order to take care of all the sodiums that are entering into the cell. And this is why aldosterone leads to an increase in sodium reabsorption and potassium secretion. Next, we're going to lump together principal cells with the function of beta intercalated cells as it relates to potassium because both secrete potassium. The top graphic is the principal cell. Bottom graphic is the type B or the beta intercalated cell. We've already talked about how potassium is moving in the principal cell, but a quick summary is potassium enters the cell across the basolateral membrane on the left, apical membrane is on the right, using the sodium-potassium ATPase, and then its fate is to leak back out those ROMK channels. There is another potassium leak channel, and it's called the BK, or what we call the big K channel. I don't necessarily need you to memorize that, but you will sometimes see them on graphics as you're studying or maybe taking reviews for board prep or something like that. You'll come across a mention of the BK channel, but we just don't need to go into that right now. Regardless, both of them allow for leakage of the potassium into the tubular lumen and on its way to the bladder. Let's move down to the type B or the beta intercalated cell. Now this is going to get into the movement of hydrogen and bicarbonate and a number of kind of complex acid-base physiology mechanisms that, again, is going to be covered in a separate standalone lecture. That's how important and a little complicated it can get. So let's not focus on that yet. Focus on the fate of potassium in this cell type. Potassium, again, is secreted, so it's going to move from the basolateral side on the left towards the apical side on the right. On the basolateral side, it enters the cell using a hydrogen potassium ATPase. So for every potassium that enters, a hydrogen is pumped out. And then the fate of this potassium is to again leak out potassium channels. And essentially you get the same effect as what was seen in the principal cell. So on the apical side, it uses potassium channels. Next, let's look at alpha intercalated cells as they relate to their function reabsorbing potassium. Again, there are complex movements of hydrogen and bicarbonate that are related to acid-base physiology that take place in the alpha intercalated cell. We're not going to go into that here. I want you to focus for now only on the fate or the movement of potassium. Now you might notice that there are some similarities to the beta intercalated cell. Let's get oriented first. The blood side is on the left, meaning this is the basolateral side on the left. Tubular lumen is on the right. So the apical side is on the right. Reabsorption means that we are going to move something from the tubular lumen back into the blood. In this case, let's think about potassium first. Potassium, again, enters the cell on the apical side using a hydrogen potassium ATPase. Now you might think to yourself, that sounds familiar. And that's because on the previous slide when we were looking at the beta intercalated cells, we also mentioned the exact same ATPase. And if you put those slides side by side, you'll notice they are almost mirror images of each other. And they're backwards. In the alpha intercalated cell, the hydrogen potassium ATPase is expressed on the apical membrane. And then the basolateral side, in contrast, has the K channel. So essentially, it's just reversed. So here, if we have potassium and we need to reabsorb it, it moves into the cell, crosses the cell, and then leaves the basolateral membrane via potassium channels and gets back in the blood again. This is the last slide that has to do with learning objective 1. And this is essentially putting it all together on one graphic of major transport mechanisms through the nephron. Over here on the left is the thick ascending limb. This is a graphic depicting everything that you just got done learning. There are some references to proteins like this one and this one. I don't need you to learn about that yet. You will learn about those when it comes to some very nuanced pathologies later on. And they are the Bartter syndrome or Barton syndrome, which involves this Bartter channel, and so on. You just don't need to know that yet. So don't let that throw you off. But here is the NKCC2, the fate of sodium, the fate of potassium, and the fate of chloride. And then calcium and magnesium are moved through a paracellular route in between cells due to the buildup of positive charge. Okay, that takes care of the thick ascending limb. In the proximal tubule, here are all of those buddy systems. This is again, it's really big on the buddy system, and that is where sodium gets reabsorbed with a buddy in all of these, except for in the case of sodium hydrogen, which is in exchange. Once all these things get inside the cell, if it is sodium, it is pumped out of the cell using the sodium potassium ATPase, which is here. And then everything else essentially moves out of the cell using its own transporter. So there you go. That kind of takes care of all the proximal convoluted tubules. The thick ascending limb we already talked about. The distal convoluted tubule comes up next. Here is the sodium chloride channel, so the NCC channel. So sodium chloride enter, and then the sodium gets pumped out using sodium potassium ATPase. Chloride gets pumped out using a chloride channel. Here is that TRP channel. So this is TRP TRPV5, which allows for calcium to get reabsorbed. Calcium gets reabsorbed and then moves out of the cell using a couple of different ways, but the main thing I really want you to know is the TRPV5 part. Going into the later portion of the distal tubule, and that is either the late distal tubule or the collecting duct, we have principal cells and then alpha or beta intercalated cells. Principal cells first are up, and we have this epithelial sodium channel right here. So sodium enters the cell, and then it leaves the cell using sodium potassium ATPase. Meanwhile, when potassium was pumped in using sodium potassium ATPase, it leaves using those potassium channels. Or it can kind of exit back out. It's a similar function to what we learned earlier there in the thick ascending limb regarding the fate of potassium. It can go either way, but for the most part, potassium is secreted that way. So if this confuses you, just kind of scratch it off for now. In the intercalated cells, the alpha intercalated cells, for now, for my purposes, don't let the hydrogens and bicarbonates confuse you. Just focus on potassium for now. Potassium is going to move into the cell using the sodium hydrogen, or sorry, the potassium hydrogen exchanger. Okay. And then in the beta intercalated cells, the exact opposite should be expressed, which it is not drawn on this image. So you would add a little arrow there for the potassium. So potassium hydrogen exchanger, and then that's essentially a summary of everything that we've covered. You can now look at these graphics that have these really busy circles and pores and channels on them, and hopefully they make a little bit of sense. My recommendation to study this part is to draw out as much as you can. Use the DSA when it comes to the details of exactly what the names of the stuff are that involves the movement. I really highly recommend that you memorize that table about water and potassium and hydrogen and bicarbonate, all that has to do with the principal cells and the intercalated cells. I really recommend that you memorize that. That'll just help you keep things straight as you go along. And then we also have the action of hormones. We have the actions of aldosterone, and aldosterone is going to work on the principal cells via stimulation of the ENaX and the sodium potassium ATPase. And then ADH, which we haven't gotten to yet, we'll talk about that later.